So, what is gene expression? Well, it's a process, okay, that has two big steps. Okay, when a gene is expressed, means the protein or the product of that gene is going to be produced in a certain amount, and it has two steps. We start with the transcription of the DNA that happens in the nucleus of the cells. It's like if you want to cook something and you have a book, okay, with different recipes. Maybe you don't want to take the book to the kitchen; it gets dirty. Maybe you want to make a picture of one of the pages or a photocopy, and you take that picture or that photocopy to the kitchen. More or less the same thing happens in the cells. We open the DNA, it's a double stranded molecule. We separate those strands, and use one as the template, and make a little copy of that tiny section, that is the gene. Okay, that copy of the gene in the form of RNA, okay, is the one that will travel to the cytoplasm, rough endoplasmic reticulum, or to the ribosomes that are freely floating in the cytoplasm. And that RNA is going to be read by the ribosomes, and depending on the sequence of nucleotides in that RNA, we are going to create a protein by simply adding different amino acids together, matching the sequence of nucleotides that appear in the RNA that, as I said, is a copy made of the DNA. That way the information travels from the DNA to the RNA to a protein, and that is simply the expression of a gene. Now, this gene expression has a very important role in what we call cell differentiation. Okay, you know, and you have studied, I'm sure before, how cells divide. Okay, cells divide by meiosis or mitosis. Mitosis, you obtain two identical cells. Now, cell differentiation is when you obtain different cells. Okay, the name is self-explanatory, or self-defining. You obtain two different cells from the parent cell. And that is the way that we obtain more than 200 types of cells that we have in the body from a single cell, that is the fertilized oocyte or the zygote, which is our first cell. Okay, and the differentiation occurs because different cells will have a different set of genes turned off or on depending on the function of that cell. Okay, we are going to see several examples of cell differentiation during physiology. Okay, and here we have some. You can take, for example, the pancreas. The beta cells produce insulin. They don't produce glucagon. And the alpha cells produce glucagon and not insulin. That means different genes are on or off in those cells. And there are different mechanisms, okay, that regulate that, turning on or off those genes. Okay, those appear in the notes, and let me tell you something very important. In the notes, sometimes you have clarifications, and I try to give more information so you can go deeper or understand what appears in the slides. That is useful to understand. You don't have to study that. Okay, in the notes you will have the acetylation, histone modification, all of these processes that turn on or off the genes, but it's not for you to study. Okay, it's not necessary now. Maybe in the future, okay, when we study some diseases that have to do with this, maybe, okay, we are going to explain that and we are going to go deeper on those, but not now. So there you have the two steps of gene expression. Notice the transcription process in the nucleus. Okay, we have here, okay, this enzyme that is separating the two strands of the DNA, making the molecule of RNA. In this case, it's called messenger RNA. And that RNA goes to the cytoplasm to be translated by the ribosomes that will make proteins, okay, after reading the sequence of the nucleotides in that messenger RNA.